Thanks, Sarah. Thanks for the invitation. Thanks for the opportunity to share with you. Can you hear me okay? Share with you some of our data in the lab over the last few years to try to begin to use neuroimaging as a biomarker for those of you who do treatment for which we can then use to improve treatment outcomes. So let me begin by asking the question, why are we doing what we're doing? Why do we need a biomarker, and why do I think we need it right away? There are no clinically validated biomarkers for addiction, and because of that, we really can't assess the addiction liability of a potential substance. We cannot assess the current severity. We can't measure blood pressure and know how addicted you are, if you will. We can't, at this point, fractionate the phenotype, although the work of Damien Fair and others are really making a huge impact in that direction. When we can begin to fractionate that phenotype, hopefully we can start to do personalized medicine and individualized treatment. And then ultimately, the question is, when do you stop? When is the person better? Can we begin to determine an efficacy of our treatment rather than simply waiting for the individual to relapse? So I would posit to you, if ultimately, if you can't measure it, how do we fix it? And we're not doing, of course, a very good job in fixing addiction. And so, for example, when I just take a look at the U.S. smoking prevalence and outcomes over the last couple of years, as of a few years ago, about 70% of all U.S. smokers wanted to stop smoking. About half of those had made a quit attempt in the last year. We're down from about half of the U.S. population smoking in the 1950s to a very resistant, about 1 in 5 still remaining smoking. But despite that, and despite these individuals going into treatment, our outcomes are really pretty poor. Even if you measure something as non-relevant in my mind as 30 days, you really still have about 2 in 5 individuals that are able to maintain abstinence. When you go out a year, it's pretty dismal. So why is it? Why is it that addiction is so resistant to treatment? And I would posit that the bottom line is that we really don't understand the neuroplastic consequences of addiction. We don't understand what that addicted brain looks like. If that's the case, how would you measure neuroplasticity directly in the humans when we can't suck out brain tissue as we can in preclinical models? Well, there are two basic approaches. You've heard a little bit about this already from Rita and from Damien. And one option is to identify basic state trait differences, alterations, if you will, within specific systems that have been hypothesized to underlie addiction. Attentional mechanisms, inhibitory control, reward processes that you heard about from Rita. And these would be indirect, if you would, biomarkers of addiction. And the second way would be to directly measure the underlying circuitry. You heard some of this from Damien from a different perspective. And if we can identify these key brain circuits and networks that underlie addiction, the hypothesis would be that these would provide more concrete, direct biomarkers of the disease. So if that's the case, where do you begin? The brain's a complicated structure. So fortunately, we have some theoretical understanding of the brain. Rita presented her ERISA hypothesis. We're tending to base our lab based on some of the preclinical work, mostly that's come from rodent models, that here are summarized by George Koob and Nora Volkoff, separating this circuitry, this underlying circuitry, into the three main components, if you will, that we think about from addiction: the initiation phase, the reward processes, the more withdrawal, negative affective processes, and craving. And as you know, for the most part, we never study these individuals. We tend to study these individuals in the lab. And so basically the overriding theme is to examine the linkages between these aberrant neuronal activity within circumscribed regions and circuits, and the dysregulated functional interactions between distributed networks that make up the withdrawal syndrome. And I'm going to use, to a great extent, nicotine as an exemplar addiction, but I'll show you some data right at the end to suggest that the data generalize to other addictions as well. Okay, so we're going to emphasize today these two other aspects and this circuitry that's been posited, despite the fact that there's human brains here, most of these data come from preclinical models, and we're using that, as I said, as a theoretical framework to test this in humans. Okay, so fortunately I followed Damien's talk, and so I'm going to actually blow through this slide, but he very nicely explained to you functional connectivity. This is our primary tool of measure as well. And let me just emphasize just a few of the things and not describe in detail what Damien already very nicely emphasized to you. This is the synchronous low-frequency activity between brain structures. And as he's shown you, it's thought to reflect the functional relationship based upon some anatomic substrate, but the functional relationship between brain regions. I can nicely blow through this. Damon showed you this already, but just importantly, and he emphasized this also in his preclinical aspects, what's nice about these networks is they co-vary during sleep and anesthesia, so we can in fact use them for cross-species comparisons. They're very reproducible all across time, so that's really nice if you're going to do a longitudinal study. They seem to be spatially consistent across subjects, so perhaps we can use these diagnostically. And these resting state networks seem to reflect the full set of what we'll call the functional networks utilized by the brain when it's actually working. A very nice paper by Steve Smith a number of years ago in PNAS completely convinced me, at least, that this sort of Lego block hypothesis of the brain, that we have these basic components in the brain. At rest, they talk to each other much the same way as these networks talk to each other when you're actually doing something. And we think this may be useful in disease. Okay, so Damien also nicely introduced the concept that the brain is organized into large-scale brain networks that have been implicated in various pathologies. And let me just emphasize three of the networks that I'm going to be talking about today, and that's the so-called default mode network, comprised specifically of the posterior cingulate, the medial prefrontal cortex, the parahippocampal gyrus. And just know that the functions that have been attributed to the default mode network includes rumination, working memory retrieval, if you will, self-reflection, planning, time travel that we do as humans. The so-called executive control network, the yin against the yang in this, that is counter to the default mode network, generally thought to be involved in externally oriented, attentionally demanding, hopefully what you're mostly doing right now, semi-paying attention to me. This is anti-correlated with the DMN and mostly the dorsolateral prefrontal cortex, that's Tony's target for his RTMS, and the parietal and medial temporal lobes. And then a network that I'm going to sort of be emphasizing a lot today, the so-called salience network, comprised of both the anterior insula and the dorsal anterior cingulate, involved in we think, conscious awareness, interoception, salience detection. Oh, I could have had a V8 kind of a thing. The butterflies that you know of error detection and awareness. Okay, so it was a nice paper, a review paper that came out just a few months ago by Jonathan Downer and Colleen Hanlon, their student Dunlop. And one can argue a bit about the way they have the trajectory, but from a more global standpoint, it's really nice to see that they've thought about how networks can begin interacting as one has basic motivational thoughts to ultimately, as we engage these networks, to finally having a behavior, a motor action. And I'd emphasize that these structures, that these networks comprise, at least in part, these three networks that I'm going to be emphasizing for you today. Okay, drilling down just a little bit then, starting from this basic preclinical model, I just want to emphasize the way that Suzanne Haber and Brian Knudsen sort of reviewed this cortico-striatal limbic circuitry, and then applying this back into addiction. And they very nicely have color-coded, Suzanne does this better than anybody else I know, color-coded these striatal regions with their major cortical components. And I'm just going to put some function on top of these for you that's relevant again to addiction. And so we think of the ventral medial PFC involved in value and decision making, reward in the OFC, error detection, as I just mentioned, in the dorsal ACC and monitoring, executive control, not on this graph, but I have to add attention and attentional mechanisms that Rita emphasized a few minutes ago, and its posterior parietal cortical interactions. And then zooming out even farther from these circuits, let's put together these three basic networks that seem to then reflect these very specific cortico-striatal interactions in the disease. Okay, so let's begin with craving. So we did a study talking about the old days. You can't see this, fortunately, because this was in 2000. This was, I think, the first or second study of using imaging to study craving in humans. And we identified a number of regions in that study. But what's really kind of humbling and cool is to go back to data that you published years ago and see if it was actually right, if it's been instantiated over the years by you and others, and or do you just want to kind of bury it and say, boy, was that we were really stupid in those days. But pointing out the regions that we demonstrated from specific cocaine cues versus neutral cues, they absolutely line up beautifully with this hypothesis that I'm trying to push to you today, and that's the interaction between these large-scale networks. So go figure. Okay, so presumably then reflecting the cognitive and affective processes that participate in cue-induced craving. So okay, how can we use resting state network dynamics to predict, in this case, smoking abstinence and its associated cognitive and affective deficits? So again, let's now take a look at withdrawal aspects, negative affect, and craving. So a number of years ago, 3 postdocs in the lab and I posited a model of how we could think about the relationship between these large-scale brain networks in addiction, and again using nicotine as an exemplar. And so what Matt Sutherland and Vani Paridath, Meredith McHugh, and I hypothesized was that individuals who were smokers in their sated state when they were smoking, the so-called salience network biased the individual towards the executive control, exteroceptive processing, and away from internal ruminations. However, during abstinence, that same salience network now biased the individual to interoception, to ruminations, to craving. And so the salience network served, if you will, as sort of the pivot of a teeter-totter, mostly shifting us exteroceptively, if you will, or interoceptively. And so the hypothesis was the interaction between these networks would be altered between smoking and abstinence and would predict withdrawal behavior. So this was a model. This was just a hypothesized model back in 2012. And so together with my collaborator, Karen Lerman at the University of Pennsylvania, we wanted to test that model. So we scanned smokers on and off smoking, and then wanted to ask the question about the interaction between these large-scale networks consistent with that model. And so all the calculations were done by Honggu and her research assistant at the time. And we created a mathematical model, an index, a single number, because after all, clinicians want a number. It's the number 12. And just make it 12, right? Is it 220? Is it 110? And so we made up a construct that we called the resource allocation index. We made that up, but mathematically it's just M here. And it's the relationship between the correlation between these 3 networks. And when we did that and simply looked at smokers on and off smoking, we saw in fact a reduction in this index between these three networks, both. This just breaks it down into left and right side of the executive control network. So there was a reduction in the relationship between these networks. And when we did the full ANOVA, that reduction in this RAI index was based on a weaker inhibition, if you will, between of the default mode network as a function of the salience network. And that was what was responsible for this weaker index. Okay? So that's nice, but who cares? Is it correlated with the behavioral changes that we see in withdrawal? And what I'm plotting for you here is a change in the index between sated and abstinent as a function of a change in the withdrawal scores. And so there's a direct relationship then between the change in the relationship across these networks and the change in craving. Okay, so that's nice. Well, does it correlate also with the cognitive deficits? And again, it does. We had these individuals did a working memory task, and when I'm simply taking a look now at changes in the signal in DLPFC, I believe this was, in VMPFC and the PCC during a working memory task. And so again, the delta of the signal between abstinence and satiety correlated very nicely with the delta in the RAI. So it looks like the empirical evidence seems to support this tripartite network. In the nicotine-deprived state, the salience network appears to shift the allocation of attentional resources to attend to these abstinence-induced cravings and leading to the enhanced DMN activity. This weaker coupling to executive control seems to, during abstinence, effectively then decreases cognitive processing. And the combination of this decreased ECN activity and less suppression of the default mode, the intrusive thoughts now start to become enhanced, may decrease this signal to noise, if you will, during performance of demanding cognitive tasks, and we wind up with the outcome measure of cognitive deficits. And we think that this reduction in executive control activity may signify this difficulty in exerting top-down control against to resist craving and thereby increase relapse. So that was a nice way that we thought that initially, actually, again, scarily demonstrated, or at least instantiated, the model, this tripartite model. And the question that we're now trying to answer is, that's nice, but is it also useful? Is it a target for us in looking at treatment outcome? And that's underway. Okay, so let's zoom down now within this salience network to one of its main components, and that's the insula. And the insula has gotten a lot of attention, of course, in the addiction field, starting with this classic paper coming in Science about 10 years ago now from Antoine Bechara's lab, demonstrating that individuals who recovered from stroke that was limited to or incorporated the insula spontaneously stopped smoking. And what's really brilliant in this paper is the insight, and there's one sentence when they asked individuals post hoc, how did you stop smoking? And their answer was, it's as if my body forgot I was a smoker. It's a great sentence in that paper. Okay, so let's talk about this insula then as the pivot point in our tripartite model. And if you haven't read, by the way, this really nice review paper by Bud Craig a number of years ago, he started to dissociate the insula into its component parts. And he talks about a gradient in the insula from posterior, you might not be able to see this back there, but from posterior insula to anterior insula, where the posterior regions are more involved in homeostatic processes and primary somatosensory interoception, moving towards the anterior regions of the insula more involved in affective and hedonic components. What I'm pointing out to you, perhaps more involved in the salience network. So a number of years ago, using graph theory, using modularity, the insula was actually divided into components, into three subcomponents. more consistent with Bud Craig's anatomic and functional postulation. And so the question then is, how does connectivity from each of these subregions of the insula differ as a function of nicotine withdrawal, and do these circuits have behavioral consequences? So this is work of John Fedota, a current postdoc in the lab, sitting over here on the left. And John asked a very targeted, hypothesis-driven question. And he said, okay, let's take these three regions of the insula as postulated functionally, and how do they talk to each of these three networks that we've hypothesized involved in addiction, and how does that differ between abstinence and satiety? So that was the model, okay? And what he found, basically, after a lot of computations, was that of these three divisions, the dorsal insula, the ventral insula, and the posterior insula, there were only 4 circuits that differentiated between satiety and abstinence. And that was between the dorsal insula and a region within the executive control network, between the ventral insula and regions within the salience network, and the posterior insula and regions within the default mode network. And then his question was, that's nice, but is it relevant? Does it have behavioral significance? And the answer was yes, that 2 of these circuits, between the ventral insula and the executive control network, very nicely and strongly predicted withdrawal symptoms, as did the circuitry between the posterior insula and the salience network, positively correlating with withdrawal. And so having the ability to follow Bud Craig's thoughts about postulating the insula, John was able to demonstrate very specific interactions between these ventral, posterior, and dorsal aspects of the insula, and how these differed as a function of the acute, this was 48 hours abstinence, of the acute abstinence syndrome, and then positing that this ventral circuit might in fact, this ventral insula, the ECN circuit, might in fact be a potential brain-based biomarker and a target potentially for treatment. And so that's the next step, and he's in the process of running a very extensive longitudinal treatment study where we're following individuals over a year period to try to see if in fact this is predictive of outcome. Okay, so one of the things that I don't think Damien talked about is something called dynamic functional connectivity. And so the assumption in resting state functional connectivity is that you're going to measure the individual not doing anything in the magnet, hopefully not moving, for some period of time, 5 minutes, 10 minutes. And then you're going to take that noise, that time signal, and you're going to regress that against the entire brain. So think about that for a second. That makes the assumption that during that 10-minute period, the individual is doing one thing. Okay? Because I'm taking the entire time series and I'm regressing it as if it's a snapshot against the entire brain's time series. Well, in fact, you're not completely listening to me right now. You're thinking maybe you're getting hungry, it's almost time for lunch, is this guy ever going to stop talking? You're coming in and out. You're not in a constant state. So is it fair to, in fact, look at this as if it was a static, that at rest, you're only doing one thing? And so there's a very new emerging construct called dynamic functional connectivity. And in contrast to this static functional connectivity that you've heard from me and from Damien, and that I'll come back to in a minute, this says, well, let's see what's the time varying aspect within rest? How is the individual and how are these regions talking to each other as a function of time? Let's make a movie of that. And one way to analyze that would be a simple sliding window and do a connectivity, let's say for the first minute, slide the window over by 1 TR, by 2 seconds, do another connectivity, slide it over by 2 seconds, do another connectivity. And what you wind up with is a matrix of interactions between structures. Got it? Okay. So you can then do some kind of a clustering analysis, and you can ask the question, so how many states can a human being actually be in? How many ways can we be cycling? Is it infinite or is it finite? And the answer is someplace between 4 and 7, okay, based on what studies you look at. So in the study that I just showed you that John did, looking at static connectivity across these 3 subregions, we then submitted the same data to a dynamic analysis, and we did what's called a silhouette analysis, and the answer in this case was 6. Okay, I don't know. So 6 different states that individuals can cycle between. Right? And then we asked the question between these. This is a matrix of the 6 states against 6 states, and then the difference between individuals between when they were sated and when they were withdrawn. And if we look at the probability to remain in a state, okay, as opposed to fluctuating, the probability to remain in that state increased as a function of abstinence. The individuals became less cognitively flexible, right? They presumably then relating to their decreased executive control. And this is interesting because the insula has been shown to be one of the most dynamic regions of the brain. But in fact, during withdrawal, there's an increase in probability of getting locked in a state, maybe reflecting, and we don't know what these states refer to, right? But they seem to be distinct, at least mathematically. Okay. So let's back up and take a look at the dependence trajectory. And so I alluded to the fact that we tend not to study people during the development of dependence, but more likely we study them during the maintenance. And so that's more associated with the negative emotional states that one sees during withdrawal. And when we think about negative emotional states, we think about the extended amygdala, the so-called dark side of addiction, if you will. And I've already introduced the concept that the insula seems to track the homeostatically relevant body sensations. And the hypothesis then is that it then alters circuit level interactions interconnecting the amygdala and regions involved in affect and motivation. So the question then is, how are withdrawal-induced symptoms reflected in insula, amygdala, cortical circuits, and how might they potentially serve as therapeutic interventions? Okay, so we're going to try to put that together from the withdrawal syndrome. So these are data that were originally collected by Matt Sutherland, at the time a postdoc in the lab, now an assistant professor down in Miami. And so he identified the amygdala in smokers and non-smokers using a very simple emotional faces task. We're not going to talk about that right now. But using the amygdala then that was identified from a functional protocol, he then used this as a seed and asked a whole brain question: who's talking to the amygdala, and how does that differ as a function of smokers versus non-smokers, on versus off smoking? Okay. And the answer was, from that seed in the amygdala, the answer was the insula, okay, as well as a somatosensory cortex. And the strength then between the amygdala and insula circuits was increased in abstinent smokers, but no effect in non-smokers. Okay. So now we've gone from the amygdala to the insula. What if we seeded the insula? Who is it talking to? Can we walk the circuit, if you will? And the answer is, when we seeded the insula, we got the default mode network. Okay? So amygdala to insula to default mode network, where the connection between the insula and the default mode circuit, again, in this case, was increased in abstinent smokers and no effect in non-smokers. So does this circuit have clinical implications again? Remember, we're doing resting state. People sitting lying in a magnet doing nothing. Why would this be relevant to the disease that we're trying to study? So we answered that question in one particular way. We made conjunction maps, and we took a look at regions where the connectivity with an insula seed was both moderated by two reasonably efficacious pharmacotherapies, and then covaried with withdrawal symptoms, right? Or covaried with the task, this amygdala task. And it turned out, when we asked that question, we got basically the same network as we did when we did it as a pure seed. Okay, so symptomatology, behavior gave us the same circuits as resting did. Okay, so then Matt wanted to ask, well, is there a relationship between any of these components and craving? And so the hypothesis was that alexithymia, the inability to report one's feelings, would predict craving, and the answer was it does. But the question then was, are there brain structures that mediate is the word I missed, and so we did a full mediation analysis to ask, starting with an insula seed, could we identify regions in the brain that mediated the relationship between alexithymia and craving? And it turned out to be from the insula to ventral medial PFC, very nicely predicted outcome. And these are individuals that are not alexithymic. You know that scale goes to about 100. These are the walking wounded. These are people up to about 50 or 60 on the alexithymia scale. Okay, so the reduction of this insula-DMN connectivity may prepare the individual, if you will, to respond to and alleviate withdrawal. It may underlie the ability of nicotine to reduce withdrawal symptoms. And these smokers with higher alexithymia may receive benefit therapeutically from interventions that emphasize emotion regulation and craving mitigation. So here's the circuit that Matt identified back in 2013. And it's always nice to know, again, is this generalized? Was there something weird about the subjects we studied at NIDA? And so he did a meta-analysis of regions that responded either to nicotinic receptor agonists or smoking cues, and really nicely overlaid beautifully with the circuits that he identified using resting state connectivity. So that came out a couple of years ago. Okay, so still with me? A couple more minutes. Lunch is coming, promise. So do these dysregulated circuits get modified by smoking? If so, where? Or are they predispositionally different, right? The question that's really difficult, if not impossible to answer using cross-sectional studies. And Rita very nicely pointed that out with some of her behavioral and circuit differences. So we took a bit of a different approach to answering that question. And it was a combined genetic imaging approach. And this was a paper that just came out in Biological a few months ago with my collaborator Rachel Tyndale in Toronto, and Sufang Li did the calculations. So there's an enzyme in the liver that metabolizes 80%-ish of nicotine into its metabolites into cotinine. It's a cytochrome P450. And it turns out that this enzyme is genetically prescribed. There's about 20-something isoforms of this enzyme. And the CYP2A6 gene, then one of the things that Rachel and her colleagues have been able to do over the years is separate individuals into so-called normal metabolizers and slow metabolizers, and then some folks that are in between. And so, as you would predict, as individuals, as we look at the change from in metabolizers from normal to slow, there's a change in that nicotine metabolism ratio, which it should be. Not very interesting from an addiction standpoint, but what blew me away a couple of years ago was the fact that this change in this nicotine metabolism changed the number of cigarettes and the phagostrum of these individuals. So here's a liver enzyme, not a liver or gene driving liver metabolism, not a gene driving brain, okay, that seems to be changing behavior. Well, if it's changing smoking behavior, smoking addiction, then it must be changing brain, okay? So we wanted to then ask the question, does CYP2A6 actually shape brain circuits? And that's a question that hadn't been asked before. So basically, can we take the gene, interact with environment, does it change brain circuits, and then does it subsequently change function? And the hypothesis was that the genotype would shape brain circuits differentially in smokers and non-smokers, which then would be reflected by changes in connectivity and brain activity. So we did an ANOVA. We had smokers and non-smokers and 3 genotypes, so it's a 2x3 ANOVA, okay? Do a whole brain analysis. And the analysis that we used in this case was a graph theory metric. You heard about that nicely from Damien. And what we used as our metric is something called functional connectivity strength. This is a way to summarize again graph theory to ask, who are the nodes? Where's the railroad stations? Are there areas in the brain that are more connected to other areas in the brain than any other areas in the brain? Okay? So this FCS is our measure then of connectivity, and we do a whole brain 2x3 ANOVA. And the answer was the dorsal anterior cingulate and the ventral striatum. Those two areas, now when we extracted the data, so these were more hubby, okay, more highly connected. And they, as you would expect, for a liver enzyme that had no nicotine to metabolize in non-smokers, there was no effect of genotype in these areas in non-smokers, but there was, in fact, only in smokers. So a classic gene by environment interaction. And more importantly, when we extracted those data, it turned out that the strength of connectivity in these two areas predicted Fagerstrom. Okay? So the less hubby you were, the more addicted you were. Still with me? So then the question was, oh, okay. So again, I always learned in graduate school, never repeat a successful experiment, but we did go back, publish it, and move on, right? But we went back to some of the work in our own lab, and a number of years ago, we asked the question between smokers and non-smokers, and using the cingulate as a hypothesis, which areas of the cingulate predicted, connectivity with the cingulate predicted addiction severity, and the answer was the ventral striatum. When we put patches on these people, it didn't change the relationship, and so we hypothesized this dorsal ACC to ventral striatum circuit was an addiction circuit. It reflected trait, not state addiction. And then a year after that, we published a paper that showed a genotypic relationship in this very same regions, in this case using the alpha-5 nicotinic receptor polymorphism. So a circuit identified using a phenotype, a circuit identified using a genotype, a brain genotype, was essentially the same as the hubs that we identified now using graph theory. Beginning to believe the data, right? It's the same regions in very different structures and very different mechanisms over the years. So this gene by environment interaction, the question then was, if you're more hubby, who's making you more hubby? Who are you talking to as a function of you being more hubby? And so then we extracted the data from these two hubs, the dorsal ACC and the ventral striatum, and we did a separate connectivity analysis and asked the question, who are you talking to? And it turned out to be the insula. And when we took the ventral striatum and did the same analysis, it turned out to be the insula. Okay? So it's the insula connectivity into these two regions that made these regions more hubby. So, does the CYP2A6 modify circuits? The answer is yes. Is it relevant? So now we wanted to probe these two regions, the ventral striatum and the dorsal ACC. In order to probe the ventral striatum, we used the now very well used monetary incentive delay task. It's a simple reward task, and simply asks the question, which areas of the brain are more involved in a reward expectation and an outcome? In a reward. I'm only going to look at the anticipation aspect of the reward task. And when we analyze the data in this task, not as a function of the task alone, gains versus neutral, but separating it again between smokers and non-smokers and slow and fast metabolizers, we didn't have enough individuals for the intermediate, we found that there was again no effect in activation in the ventral striatum on this task in non-smokers, but only in smokers. We then put a patch on these individuals and try to make them better. That's okay. You can hear me, right? Mostly? Right, there we go. Okay. And so we scanned them under a placebo condition, under acute abstinence, and then scanned them and then scanned them with a patch. Oh, I should say these data again replicated a study we had published a few years ago, just looking at smokers versus non-smokers. Nice. But now we put a patch on these individuals and take a look what it did to both actually these slow metabolizers, both smokers and non-smokers, and it increased the activation. So nicotine actually made the response in these individuals bigger. I don't know if it was better, but it was bigger, and it was true for both smokers and non-smokers. Okay. Well, what about the dorsal ACC? So we know a nice way to activate that, and that's with a very simple response inhibition task, a go-no-go task. So we took that mask out, and again, reanalyzed our data as a function of gene by environment, smokers and non-smokers, and got essentially the same answer, and scanned these guys again with a nicotine patch, and it normalized. So it appears then that this gene by environment interactions on connectivity were located within the cortico-striatal circuits that previously been implicated in nicotine addiction. I didn't show you the data. I'm not showing you the data that there was no difference in gray matter density in these individuals. And these circuits are modified only in smokers. If that's the case, that would suggest that it's the relative difference in the concentration of nicotine, because we're changing metabolism only, but the relative concentration of nicotine that presumably gets into the brain that induces these changes. Okay? Not correlated, perhaps. Rita's not sure. So the same gene by environment pattern seen at rest and task suggests the functional significance to these circuits. This insular, again, dorsal ACC striatal circuit's part of the salience network that I've been talking to you about, and that smokers with the slower genotype, relatively greater concentrations of nicotine in the brain, and this is why I think what I do, Rita, they're less responsive to anticipation of gains, they're less responsive to errors, and for those of you who do treatment, I think that has a lot of implications then in this plasticity that's very specific. This was, remember, a whole brain, agnostic, data-driven approach, and only these two areas came up as being less hubby. And then, of course, back to the question at hand, can these circuits serve as biomarker targets for treatment? And again, something that we're in the process of doing these painful longitudinal studies. Okay, last piece of empirical data. I've been using nicotine as an exemplar addiction, and I promise that we're going to take a look at that as a function of another addiction. Do these circuits generalize to cocaine dependence? And so this was a study led by Xia Liang just a few years ago in the lab. And she identified circuits, in this case not using so-called independent component analysis, but graph theory. She used something called modularity analysis, and again identified these modules that are essentially the same as the components in an independent component analysis. And we have the same default mode, salience network, executive control network. In this case, she also identified and we analyzed medial temporal lobe and a striatal module. Okay? So these basic 5 modules, and she wanted to simply ask the question, how do these modules differ, looking at this matrix now, between healthy controls and cocaine-dependent individuals? And when you do the math and look at the difference between the two, it turned out to be the default mode to salience network relationship that was different between healthy controls and cocaine dependence. And if you can scratch your head and think back 30 minutes ago or so, it's exactly the same thing that we found in nicotine-dependent individuals using a completely different cohort and a completely different way of analyzing the data. Okay? So, in conclusion, what I tried to show you today was that resting state connectivity can, in fact, identify functionally connected brain circuits that reflect addiction, both state and trait. And what we need to do now, and what we're in the process of doing in my lab and a number of other labs, is to identify which of these circuits are in fact plastic. We think we have some hint at that using the CYP2A6 data. Do they change? And Rita showed you a little bit of that data. Do they change? Do they normalize with treatment and with a treatment intervention? Perhaps what Tony was trying to show you a few minutes ago with TMS. Can we actively target these structures and causatively, not correlatively, but causatively change their structure? Can these circuits serve as biomarkers? So let me just also emphasize the fact what I've been showing you today are things that are different. And things that are different, there was a really nice review paper a year ago, 2 years ago, by John Gabrielli and his wife, Susan Whitfield Gabrielli, that emphasized that things that are different are not necessarily things that are predictors of the future. So everything I've been showing you are differences, right? We're beginning to believe some of them, right? But are they predictors? Are they useful as biomarkers? Can I give you, as clinicians, some of you in the audience, tools upon which you can use to measure against your treatment? So will they be able to quantify nicotine addiction severity? Can we individualize treatments, as Damien is really nicely trying to fractionate the phenotype, in his case in ADHD? Can we take a look at addicts for that subtype for anxiety or cognitive or reward deficits, depression, etc.? Can we begin to then think about personalizing our treatments and treat these individuals differently instead of simply saying, you're a smoker or you're a cocaine addict? Okay? And can we use them, importantly, to predict treatment efficacy? So with that, let me thank the folks in the lab. And as Tony pointed out, it takes a village. There's really a lot of folks that are necessary to do these kinds of studies. I highlighted some of them along the way, and some of my collaborators outside of NIDA. And I thank NIDA for their support, and thanks for your attention.
We have a couple minutes for questions.
Yeah.
Identified the alcohol deprivation effect, where animals that were accommodated to self-administration of alcohol and it was removed beyond any point where any withdrawal specifically would be acting, return them to access to alcohol, and they drank at much higher levels than before it was discontinued. And I think people thought this was maybe stress hormone related, but the salience network interaction with the default network seems like a much more plausible explanation. I just wonder if you thought any about the deprivation and return to access.
Yeah, no, that's a great question. And much like Damien's trying to do, we've developed, and I certainly didn't have time to talk to you about some of our preclinical models where we're trying to, in fact, again, causatively turn these rats into smokers, into nicotine-dependent individuals. So I think I don't know the study in particular that you just referred to, but it sounds like the reinstatement model that Yevin Shahem and others have been talking about I think Rita talked about the change in craving as a function of time in humans. And we know in animals, certainly that after the self-administration behavior has been instantiated and then actively withdrawn and then incubated, these rats were then put into jail, back into their home environment for a day, a week, a month, up to 3 months, I think, in the original study. The longer the rats were in jail, these were fully extinguished rats, and then exposed to cues, the quicker they were to reinstate their dependence. And so this would suggest that the plasticity occurs in the absence of the drug, right? So that incubation is happening after the self-administration, after the extinction, and in the absence of any cues and any exposure to the drug. So that plasticity perhaps is set up as a function of that self-administration, but it's not the self-administration that's causing the plasticity, putatively. It's the absence of that self-administration. It's that incubation period. And whether that is, and it's a great question in terms of then, now using modern neuroscience techniques, can we begin to look at the brain regions, the networks that are involved is the next step. And we're beginning to do that actually in a collaboration with George Koob's lab, where they're using fentanyl self-administration. And we're just now scanning his rats as a function of the cue-induced reinstatement. It's a really important question to see if this model then holds causatively. I'll tell you in a year. Yeah?
I have two questions. So you were just starting to talk about the opioids, and I was wondering if there's kind of work that's being done looking at opioid, like how it's
Yeah, there's much less data in imaging in opiate-dependent individuals. Ironically, in terms of the opioid epidemic, certainly in the States these days, we're just beginning. I just read the last version of a protocol we're about to submit to the IRB to look at opioid withdrawal actually in impaired physicians as a model, because they tend to have outcomes, right? Better outcomes than the usual opiate addict. And so if you're going to do a predictor, you need outcomes. You need people to succeed and fail. There's virtually no data on this right now.
And then just my second question is, there's a lot of polysubstances. That's more than norm, I would say. So how, in the studies that you've done, control for that?
Yeah, that's a tough question. That's really the nub. And you know, Tony talked about dual diagnosis and dual dependence. That's the reality on the ground, right? That's where we should be living. There are no smokers. There's only anxious smokers, depressed smokers, bipolar smokers, right? There's no smokers, right? And you know what I learned in day one in graduate school is keep everything constant and change one thing, right? So all right, I'm just going to look at smoking and I'm going to throw out all that other stuff, and we do that I think at our own peril. So all of the studies that I've shown you with smokers, people with any other comorbid psychiatric disorders are not studied. Okay? We do allow, of course, some drug use, but nobody with diagnostic DSM, alcohol dependence or what have you, right? So it's really the important question. It's the thing to hit, I think, going forward. I've talked to smart clinicians and asked the question, how do I study this when I have, let's say, depression and smoking? And they're dual occurring. They might be at different levels of intensity. They might both be moving somewhat differently because you're being treated for the depression, you may be the smoking abstention. How do I group these people? How do I study something where I have an interaction that I don't understand? And if you have an answer to that, please let me know. I don't know how to do that, but that's the absolute critical thing clinically, because we're just ignoring that clinical reality.